Disclosure before we start, we're changing gears right now. My background, I'm pharmacist, so I have been working in preclinical research in epilepsy for a number of years. And over the years, I really got interested about our brain clocks. So I'm going to tell you a little bit about epilepsy rhythms, so the rhythms that we can see in different groups of people with epilepsy, how actually our brain clocks are ticking. And yes, we do have a central brain clock, and most of our cells, not only in our body but also in our brain, have their own clock. So we're going to be talking a little bit about that. And we asked the question, can the disruptions of these brain clocks, internal clocks, cause trigger seizure? And then we are going to be looking and wrapping up with Melissa's talk that is going to bring a little bit more about the sleep side of things. So first of all, about epilepsy rhythms. We thought for many, many years that seizures were appearing randomly. However, now we know that they actually have a time scale. So they appear about 90% of people with epilepsy in a general way with the epilepsies, they have some sort of rhythmicity. This could be different. So some people would have multi-diem rhythmicity, that it means that seizures are more likely to happen every 20, 30 days and recur again. So other people actually have weekly cycles, or they have some rhythms that they have annual cycles. But what I'm very interested in my lab is to look at the circadian cycles. So what would be the circadian cycles? So the circadian cycles would be people that will be more likely to have seizures, either only nocturnal seizures or during the day, or have a mixed pattern. So we know that this is occurring in a number of people with epilepsy, and this is highly specific to the person. So what we don't know yet, and in spite of this being very well characterized in the clinical side over the years with long-term electroencephalographic recordings, we don't know what is actually happening inside of certain brain structures and inside of the cells that could be leading to the appearance of the seizures at certain time of the day. So we are interested in the lab looking at zooming in inside of the brain, zooming in into the cells in order to understand a bit more of these rhythms and inform us whenever we are trying to develop more precise therapeutics, and also whether there is a better time of the day for certain person to take their treatments, or also informing as regards diagnosis. So moving now to our brain master clock. I told you that we have a brain master clock in our brain, and if you can see like this very, very small region in the bottom of the brain. I know it's very difficult to see on the slide. So this is called the suprachiasmatic nucleus. It has a very big name, but we will keep it calling it the master clock. So this is our central clock in the brain. And even though on the right-hand side, the image, I'm going to show lots of scary, very complex images, but you don't need to worry about them. I will try to keep it the key message on them. So this master clock, the suprachiasmatic nucleus, is our central clock. And this central clock is ticking and sending messages to every other clock in our brain cells, and also coordinating the cells in our body. So we know that it is regulated through the light that we have, so the light and dark cycle. But in spite of the light or darkness, we still have the clock ticking. Every 24 hours, or approximately 24 hours, we're going to have a new cycle happening. We like to compare the role of the master clock as the conductor of an orchestra. So it is there synchronizing all the other clocks, and we are going to be looking a little bit more what is the role of these clocks in epilepsy. So what does the central clock or the orchestra conductor coordinates? It coordinates our circadian rhythms. So the circadian rhythms are those 24 hours rhythms in our body, and they change, they do physiological and behavioral changes every day. So things that were very well known as regards to be controlled or regulated by circadian rhythm is our sleep-awake cycle. So it's one thing that we know, that our sleep-awake cycle is controlled by our master clock. Also, we have, for instance, the times of the day that we're going to be more hungry. So this is also controlled by our circadian molecular clock. However, things that we cannot control are also controlled by this master clock. For instance, our body temperature, blood pressure, and other changes. But I'm going to leave this part, and Melissa is going to talk a little bit more as regards the sleep perspective later on. In spite of those initial examples, what else is controlled by our master clock? So I know that most of you already know about how genes work. So we have our genome, we have the DNA, and the readout from that DNA is going to be the expression of proteins, and I'm sure that you're going to hear a lot more about later on about this. However, it's already known that our central clock is able to coordinate a large portion of all these protein coding genes that we have. So it's a very powerful mechanism of coordination, and the synchronization that we need to achieve in these clocks, it's something very, very critical for us to keep the health in the brain and to keep processes that are very important stable over time. There is a big list of things that we already know that the master clock coordinate, and lots of pathways or lots of important things in this list. They are actually very important for seizures and for the development of epilepsy. But what make this clock tick? Again, here we have a diagram that shows a quite complicated idea of genes, but I'm going to try to simplify it in the sense that I would like that you would think as a seesaw. So we have two arms in this seesaw. So we have one arm that we have these genes called BMAL1 and clock. They are positive regulators. And we have on the other side of seesaw genes that are called period and cry, and they're the negative. So the balance of these genes happens every 24 hours. And that's how our master clock is ticking, and that's how our master clock will inform the time of the day for all the other brain cells and the body for things to work in a molecular way. So this process takes about 24 hours to work. And the role of light, either natural light or artificial light here, is to help to synchronize, to make sure that our internal clock is ticking on time. So moving from that, okay, this is happening in all our brains. So what happens when these clocks are not ticking or are disrupted? And we verified, not only in our group, but other groups verified, that there is a disruption in the molecular clock, for instance, here in the example of temporal lobe epilepsy. So we measured this BMAL1 gene, that was that key component for the clock to work, that is responsible for the positive side of the clock. And this gene seems to be reduced, the expression is reduced in samples resected after surgery. In our lab, we have to rely a lot to understand and to be able to zoom in the brain and in the mechanisms. We need to use mice models in order to be able to understand the processes in each cell and over time. So we took these mice that underwent the status epilepticus with the injection of a chemical into their brains, and we verified the expression of this clock regulator was also reduced in the mouse model very early after the status epilepticus. So for us, it was a proof that there is this disruption, and we need to understand a little bit more the role about this disruption and how can we actually not only understand the role of the disruption, how can we, if we can prevent or use it in the future as one possible therapeutic approach. So this work is part of the work of Dr. Rana Benvenuti, who is funded by Epilepsy Ireland as well, and I'm just bringing a little bit of the overview here. So our next question was, how much disruption of these clocks can trigger seizures? Well, to bring this part, I would like to change your attention for the main players in the brain. Whenever we talk about the brain and whenever we talk about seizures, we talk a lot about neuronal activity and the neurons as the main players. Here we decided to focus on some certain types of cells that would, I would say, be less studied for a number of years, that are called the microglial cells. They are only about 10–15% of the overall amount of cells in the brain. However, even though they are this smaller population, we know more and more about them, and that they are very important in maintaining a number of functions. For instance, the microglial cells, they could be called the guardians of the brain. So they are already very known to be responsible, for instance, doing the security and the cleaning of the brain, and thinking of that the cleaning of the brain happens a lot during the night and at sleep. We thought that they would be very interesting cell types for us to focus and understand about their clock. So we already know that they have a very strong circadian clock, and they are able to not only control inflammation in the brain, but they are involved in a number of other processes that could be very important in seizure generation and epilepsy generation. So to understand a bit what we've done next, this is actually the project of a very talented PhD student in the lab, and it took her a long time to create a mouse model where we could generate these lines with a reduction of the BMAL1 gene, the core clock gene, only in microglial cells. So, wait a minute. Now I'm telling you, it's only 10% of the cells in the brain, 10 to 15%, so it's a very small portion. And we are getting the reduction of just one gene that is responsible for the circadian rhythm inside of the microglial cells. And this is not even a full reduction of BMAL1, it's just a decrease of BMAL1 in these microglial cells. Well, I tried to explain in about 30 seconds, but this took her more than a year. So it's just an idea to give you how much it takes to characterize these lines. What we did in these mice, whenever we generated them and we noticed that they had a disruption in their circadian rhythms, they had a disruption in their sleep, even though being in such a small proportion of the brain, we looked at their behavior. We've seen that, I'm not going to go through the graphs here, I'm just going to talk through, but if you look in this diagram, the purple one, the top you have the mice that are controlled. This is a normal movement for a mouse, so they are moving in all those tracks. And in the bottom one are the mice that do not sleep properly, that they have the circadian rhythm disruption in those cells. So you can see that they move way more. So we verified that there was an increase in locomotor activity and movement in these mice. However, they seem to not have any changes in anxiety. As a next step, we looked at cognition and we look at memory using different tests that we can assess in the lab in order to see whether this disruption would change any of these parameters. And we didn't find any changes. So this mild change in the circadian rhythm was not able to change any aspect of memory and cognition. Our next question then was, okay, having this very focused small disruption in the circadian rhythm, would that make mice more susceptible to seizures? So how much actually disruption we need to have seizures? If we look at these mice and we connect them to electroencephalographic recording, over time, we did not see that they have any spontaneous seizure. However, if we take them, we give them a low dose of a chemoconvulsant called kainic acid that usually would not trigger seizure in a mouse, if we give that one single injection, we see that these mice will have seizures. So whenever we have this small disruption in the circadian rhythm, we will have mice that are more prone to have seizures while the controls are not having seizures. Also, whenever we look at these brains, the brains of these mice, we could see, and I'm not going to go through the graphs again, I don't mean to scare you, that there is already a change in the inflammatory profile in proteins that and genes that are responsible and are known to be involved in neuroinflammation and known to be involved in seizures recurrence over time. So this gives us an indication of mechanism of what could be happening at very early stages after a seizure. So just to give you a summary and where we are at, we know that the majority of people with epilepsy present some sort of rhythms in their seizure presentation. The brain clock is disrupted in human and mouse TLE, so we already know that there is certain type of molecular disruption, and even a mild disadjustment in the master clock makes the mice more prone to have seizures and inflammation. So there is this correlation between generating disruption in circadian rhythms leading to more seizures, and more seizures are likely to lead to more disruption in the rhythms. So is the brain clock disrupted in other cell types, in other types of epilepsy, sorry? We've been looking at different types of epilepsies, and it's really important to characterize and differentiate what is a circadian disorder and what is a sleep disorder. So circadian coordinates sleep, but not necessarily having a sleep disorder means that there is a disruption in the brain master clock. And we look forward to keep the research to try to adjust the clocks as a potential treatment. So I just would like to finish and thank a lot to my group. It's an amazing bunch of people, and the research is never done by one person. So all the data that they presented is part of their hard work, and with the support, large support from Epilepsy Ireland. So thank you very much.